Hello guys and welcome back. We've been talking about plant growth hormones. As promised, I'm back with another video today onwards in which we'll start talking about plant growth hormones in detail. Today's video is going to focus on the first plant growth hormone, that is auxin. Well, I hope you remember that auxin falls in the category of plant growth promoters. We also have plant growth inhibitors that include abscisic acid and ethylene that we'll discuss in the subsequent videos. This video is going to be divided into two parts. In the first part, we are going to understand how auxin was discovered. In the second part, we are going to move on and study where it is produced and what function it performs in the plant body. So let's quickly begin. The discovery of auxins was firstly observed by the very famous Charles Darwin and his son Francis Darwin in the year 1881. This observation was made in the coleoptiles of Canary grass. So what they observed was that when the canary grass plant, the tip of the coleoptile was illuminated by unidirectional light. That is, the light is coming from only one direction. In this case, they observed that the tip of the coleoptile, जो इस coleoptile का tip region है, shoot apex है, that is going to bend towards the direction of the light source, a phenomena that we commonly now know as phototropism, the response to the stimuli, that is light. So here this coleoptile, as they observed, showed a bending towards the light source. Now in another experiment, they observed that when they decapitated this shoot tip, no bending was observed towards the direction of light. So in the second experiment, it was observed when this coleoptile was removed, when it was cut off, no bending was observed. So the plant remained intact. So they deciphered from this that definitely there was some substance that was present in the tip of the shoot that was responsible for this phototropism. However, they could not discover this element yet. They just told about the presence of a such chemical. Another experiment that gave the proof of this chemical was performed by a scientist known as Wendt. Let's look at what he did. Now let's talk about the experiment performed by the scientist by the name of Wendt. It was performed in the year 1928 and he studied the Avena coleoptiles. Please remember that Charles and Francis Darwin studied the canary grass coleoptiles. He, as in Wendt, studied the Avena coleoptile. Avena is basically what we know as oat seedlings. Okay, so these are the oat seedlings on which he made his studies. So what was the experiment all about? So he took the Avena coleoptile and here we see that this coleoptile has its shoot tip intact. Now he decapitated the shoot tip, मतलब उन्होंने shoot tip को काट दी. अब जो shoot tip इन्होंने cut off करी थी, he has placed it on a agar block. What is agar? Agar is a polymer जो कि Gelerium और Gracilaria नाम के agar से organism से मिलता है. ये organisms basically एक तरह के red algae हैं. Agar is pretty much like gelatin. आपने gelatin देखा होगा ना jellies वगैरह में. It's pretty much like that. So एक agar के block पे he has now placed this cut off shoot tip. Now कुछ समय के लिए इन्होंने shoot tip को agar block पे छोड़ दिया. Now after that he has discarded the shoot tip और ये agar का जो small piece था जिसके ऊपर ये shoot tip पड़ा हुआ था उसको उन्होंने cut off कर लिया. After cutting that off, this agar piece was replaced on this Avena coleoptile जिसका shoot हमने initially cut किया था. यहाँ पे हमने shoot cut करके इसको place किया था ना, इसी Avena coleoptile पे हमने अभी एक agar का piece डाल दिया. कोलियोप्टाइल का जो टिप है वो हमने डिस्कार्ड कर दिया माइंड यू, बट हमने क्या प्लेस किया, ये अगार ब्लॉक प्लेस कर दिया. ऑन प्लेसिंग दिस अगार ब्लॉक he again observed that on illuminating with unidirectional light a bending was observed. So what did he conclude from it? He concluded कि शूट टिप में एक ऐसा केमिकल होता है जो कि अगार ब्लॉक में डिफ्यूज हो जाता है. This chemical on the shoot tip that diffuses into the agar block is responsible for the bending towards light, and therefore he gave proof that there was a substance in the shoot tip that caused the bending, and that substance we know is known as auxins. Now let's quickly move on and see the final experiment where auxin was actually isolated. Now we come to the third and final step where auxin was finally isolated and characterized. It was done by scientists Kogl and Thiemann. Kogl observed and in fact isolated auxin from the urine of a Dutch patient. See, isolated auxin from the urine of a Dutch patient. Now the auxin they finally derived as the natural form was indole 3 acetic acid that we are going to discuss in a small bit. Now let's start talking about auxins, what they are and how they work. Well, auxin word is derived from the Greek word auxein, and auxein means to grow. And definitely we know that this is a plant growth promoter. It is going to help the plant to grow. Now, where is auxin synthesized? Auxin is synthesized in the shoot and root apex, the tip of the shoot and root, as we already know and we have seen in the discovery experiments also. From these shoot and root tips, auxin migrates wherever it has to act. जहाँ भी act करना होगा वहाँ वो migrate कर जाएगा. Now, what is auxin derived from? Chemically, as we've already discussed in the previous video, it is derived from a compound known as indole. So auxins are indole derivatives. Now, what are the types of auxins? Well, auxins could be natural, that are synthesized in the plant body, and in this case we know natural plant growth hormones are known as phytohormones. An example of these phytohormones is indole 3 acetic acid and indole butyric acid. They come in the category of natural auxins and they are the phytohormones. They are made by the plant itself. Now the second category is synthetic auxins. This means that these are the auxins we have artificially manufactured, and as we've already discussed, they will not be called phytohormones. Instead, they will be called plant growth regulators, shortly abbreviated as PGRs, plant growth regulators. Auxins that fall in this category include naphthalene acetic acid and 2,4-D, that is a common herbicide. Now, where are these auxins used? Well, they are very commonly used in agriculture and horticulture practices. Horticulture is raising the flowers. Now, let's talk about what exactly these auxins do. The first function of auxin is, well, it helps in cell division, whether it be inside the plant body or whether it be outside the plant body. Outside the plant body, we have a practice known as tissue culture. What is tissue culture? Well, you take a cell from the plant body. It could be from the leaf, it could be a pollen grain, anther, anything. And then you put it in a medium that contains nutrients and also growth hormones. One of these important growth hormones is auxins. What role does auxin play? Well, auxin increases the cell division rate so that the one cell that we took would rapidly divide to form a ball of cells that we know as callus. So, auxin helps in callus formation in tissue culture. If you want to know more about tissue culture, I have a video on that. Please check that out. Now, moving on to what auxin does inside the plant body. Well, it helps in secondary growth. As we have discussed earlier, primary growth is the increase in length of the plant. Secondary growth would give the plant width. It will help the plant gain girth. So, this is done by the cambial tissue where your auxins is going to increase the cell division. Also, it helps in the differentiation of the cells That is, the meristematic cells differentiate to form what? Your xylem and phloem. In higher concentrations of auxin, usually it induces both xylem and phloem formation. In lower concentrations of auxin, only phloem formation is induced. Now let's move on to the second function. Adventitious root initiation. The normal roots that we see in the plant are the natural roots. Adventitious roots refer to false root. Let's say you have a stem cutting. There's a rose plant and you have a stem cutting and you want to grow it. So what you would normally do is you would dip it in a solution having many growth hormones. One of them is auxins. Auxins would help that stem cutting to develop new roots. The auxins used usually for the purpose are naphthalene acetic acid and indole butyric acid. So they are known to induce root formation. Now let's look at the next function. Another function of auxin is that it induces flowering but in a uniform manner. It is helpful in commercial plants like pineapple, where we want to synchronize the flowering. Another function is it prevents early drop of fruit and leaves. If the leaves are young, it's going to prevent their abscission. On the other hand, it in fact also helps in the abscission, that is the fall of old leaves and fruits. This is going to help the nutrients to be available for the new leaves. Nutrient recycling maybe auxin helps. Another feature is that it induces parthenocarpy. Well, what is parthenocarpy? Parthenocarpy is the formation of seedless fruit. So, a fruit that does not have seed. Seed is the product of fertilization. So, we want to form a fruit without the fertilization process. So, what we do is we take the flower and we spray it with auxin. On spraying, the flower would get converted and form the fruit without the real fertilization process. So the benefit would be that our fruit would not have seeds. This is beneficial in plants like tomato, where we want to exclude the seeds. Now another function is that auxin is used as a herbicide also. 2,4-D is a synthetic auxin that is sprayed in the monocot plant fields. Why in the monocot plant fields? Because it specifically targets dicot weeds. So if I have a plant that has monocot weed crop and some dicot weed has cropped up in the field, this 2,4-D is going to kill that dicot weed but let my monocot plant be unaffected. So if there's a rice field, I can go ahead and spray this 2,4-D on this. Now the last function of auxins is the function of apical dominance. Here's an important piece of information. I post my videos on Saturdays and Tuesdays. So the next part is going to be available on Tuesday. Now coming back to apical dominance. Well, apical dominance is a phenomena where the presence of the apical bud, that is the bud present at the apex, is not going to allow lateral buds to grow. What are lateral buds? Well, this is my apical bud present at the apex. The lateral buds are present on the sides of the plant. They allow the plant to broaden, to have some foliage. So where would I want some lateral growth? Let's say I have a tea plant where I don't want my tea plant to just be tall. Rather, I want it to have a spread so that I could get more number of leaves. Let's say there's a hedge that I'm going to use to make my boundary. So instead of being tall, I'd like my hedge to also have some width to look beautiful. So in such plants, what I do is go ahead and chop off this apical portion. The moment this apical portion is cut off, the amount of auxins is going to go down. Now my lateral buds would have an opportunity to grow and my plant would also have some foliage. So that was all for auxins. The next video is going to talk about another plant growth promoter, that is gibberellin. Don't forget to subscribe to my channel and like this video.